Hi everyone, my name is Cailynn and my lab is affiliated with the Naval Medical Center of San Diego as well as the Department of Psychiatry at the School of Medicine of UCSD, and I'm studying the comorbidity of mild traumatic brain injury and PTSD by using neuroimaging. I'm studying this because mild traumatic brain injury, which also known as concussion, and post-traumatic stress disorder, or PTSD, have a very high comorbidity rates in the military population. There are lots of overlap between the symptoms of PTSD and the symptoms of MTBI. For example, fatigue, irritability, and memory problems, which make it really hard to make diagnoses and provide accurate treatments. And the neural mechanism of this comorbidity remain under study. So if you take a look at the picture on the right, which depicted the cone of vulnerability, which essentially just like a triangular regions that explains why some of the brain areas are more vulnerable to rotational forces during brain injury because of the movement of the brain tissue. And this area overlap with the brain area that involves in PTSD a lot, which including amygdala, because the neurobiology of PTSD involves a rewiring of neural network, and stress induces a abnormal synaptic plasticity within the limbic system, such as the hippocampus and amygdala. So, synaptic plasticity is just a term that describes the changes at the synapse between two neurons. So, in other words, stress-induced changes of the connection between two neurons. So it just got me wonder, will mild traumatic brain injury induce any alteration at the synaptic level within the amygdala, which can play an important role in emotional regulation? And maybe this alteration can be related to the manifestation of PTSD or even maintenance of PTSD in these patients. And it has been found in literature that traumatic brain injury induces structural plasticity within the amygdala. And neurons shows dendritic remodeling, specifically like changing its morphology, its structure, that could potentially increase neural communication. And these changes are related to the anxiety-like behavior in animals. And clinical study also try to use neuroimaging to investigate the amygdala as a potential biomarker in patients with mild traumatic brain injury and emotional deficits, but producing conflicting results. So some study show that comorbid groups have a larger amygdala volume, and some studies show the opposite. So the goal of my study is just really trying to see whether there is like a biological link between the amygdala and the PTSD symptoms in patients with mild traumatic brain injury. So to answer this question, I use neuroimaging to compute a volumetric measurement of the amygdala, and I use the PTSD symptom checklist for DSM-5, which is also known as PCL-5, to quantify the severity of PTSD. By using mediation analysis, I found that patients with mild traumatic brain injury have significantly more severe PTSD symptoms, and the amygdala of these patients who have a more severe PTSD symptom show a trend of enlargement in comparison to patients who don't have PTSD symptoms. But this is not statistically significant, which differs from some of the previous study that had a larger effect size and larger sample size, which found that there are significant enlargements of amygdala, suggesting that further investigation with a larger sample size is needed. So what I'm presenting today is really just a closer look at how the alteration in the brain structure that is important in emotional regulation and emotional processing in patients with brain injury, and how it can be related to behavioral outcomes. However, the neural mechanism of this comorbidity is likely to be very complicated. Nonetheless, we learned that the amygdala has the potential to mediate development of psychiatric disorder after brain injury. Future study can examine the functional connectivity of the amygdala to the frontal cortex because the alteration is likely to be circuit-based instead of like structural-based. In neuroimaging, in conjunctions with preclinical study for microscopic alteration after TBI, is needed to untangle this complicated relationship. And finally, I would like to thank my mentor, Dr. Ann Hoffer and Dr. Gimbo, who offer both emotional and professional support generously and inspired me in many different ways throughout this whole process. This is all for my research project. Thank you so much for listening.